Class, we are going to start pigment accumulation in our cells. So, first of all, we will discuss melanin. Melanin before studying accumulation of melanin, we will discuss normal physiology and normal presence and normal production of melanin in our body. So, as you know, everyone born with a different skin color. Okay, so skin color actually defined by the melanin production, how much melanin is producing from your cells and accumulating. Then your skin color is actually is from the melanin. Okay, melanocytes, melanosomes are keywords in our today's lecture, and tyrosine as well. Okay, so first of all, we will discuss anatomy of skin. Sorry for interruption. There was a net issue. So we were studying about the layers of skin. So there are 3 layers of skin. Okay, so
screen नहीं नजर आ रही.
Thank you. So if we talk about the general anatomy of the skin, there are 3 layers of the skin. Is composed of with 3 layers. Okay, so first layer is epidermis, which is outermost layer, and second is dermis, which is actually a second layer after epidermis, and third layer is subcutaneous tissue. Okay, so these are basic layers of human skin. So if we talk about epidermis only, then we say there are different layers in the epidermis. Okay, now we are going to study just epidermis. Means we are going to study outermost layer, and if we study outermost layer of the skin, then we say there are different layers in the skin. Okay, you have studied 5 to 6 layers in the epidermis, but here you will observe a few things, few important things. Okay, so these few important things are actually like keratinocytes, melanocytes. These things are actually cells or cells granules, for example melanosomes. And keratinocytes are basic cells, then melanocytes also exist, then surface is actually outermost layer. Okay, so melanocytes release melanin, and melanin accumulates in the melanosomes. Okay, and melanosomes accumulate in the different keratinocytes in the layer. So in epidermis, accumulation of melanin occurs in melanosomes, and melanosomes accumulates into different cells. Okay, so what are the melanosomes? Melanosomes are membrane bounded structure found in the epidermal cells, melanocytes and keratinocytes. Okay, so please keep in mind, first of all, melanosomes exist in the inside the cells, and most common they exist in the melanocytes and keratinocytes. Okay, so from this diagram you can easily observe keratinocytes and melanocytes. Melanocytes is actually, here you can observe a dark cell in the bottom with multiple branches. Now melanocytes, after melanocytes you can observe keratinocytes with slightly brown color skin, normal skin cells. Okay, and within keratinocytes you can observe a membrane bounded organelle is present. This membrane bounded body is known as melanosome. Okay, so melanosomes exist in the melanocytes as well as in keratinocytes. Okay, so they are the basic unit of skin pigmentation. Means to say, melanocytes, keratinocytes, and melanosomes, which are present in the melanocytes and keratinocytes, are basic units for skin pigmentation. Okay. Now the number distribution, the number of melanosomes, level of melanosomes, and the uh, size of melanosomes determines your skin colors. Okay, in very simple wording, how much melanocytes are present, how much uh, uh, what about size of melanocytes. Okay, first of all, distribution means to say how much melanosomes are present inside the cells, and distribution or number, almost they are similar, but in distribution we usually discuss about the spread of melanosomes. Means if melanosomes are present in most of the keratinocytes, then we say pigmentation, then we say dark brown pigment will appear widespread throughout the body. Okay, so degree of skin pigmentation is determined by these three factors: number of melanosomes, distribution of melanosomes, and the size of melanosomes as well. Okay, there are two kinds. There are usually melanosome. There are usually two other kinds of melanin, primary types of melanins: eumelanin and pheomelanin. Eumelanin actually, both are actually primary melanin. Means to say, melanin, which is actually a very dark brown pigment, originate from these two forms of melanin. So you can say eumelanin actually appears dark or brown from start. Okay, but pheomelanin usually appears red in color. Okay, so these eumelanin appears dark brown in color, but pheomelanin appears red in color. So this is actually a maturation process of melanosomes. How melanosomes forms, and how and where it matures. Okay, so first of all, usually I have told you that melanosomes exist in keratinocytes as well as in melanocytes. So they get mature, and they first of all they born and mature in the keratinocytes or melanocytes. Most of the time in melanocytes. Okay, so first of all, they get detached from the rough endoplasmic reticulum. You can observe in the diagram. Then they bind with the help of tyrosinase enzyme. Okay, with the help of tyrosinase enzyme, they get mature. So in stage 2, usually they appear in very light brown in color. Okay, they appear in very light brown in color, which is stage 2 is very initial stage, and stage 1 usually there is apparently no dark brown color can be seen. So but in stage 2, very light dark pigment can observe. Okay, but in stage 3, dark brown color appear more. Okay, then which is and stage 3 and stage 4 actually is a um, melanin, actually the dark and brown pigment, and the last stage is stage 4, in which a whole cell gets accumulated with melanin, mature melanin, and this is actually a maturation of melanosomes. In melanosomes, melanin gets accumulated, and in mature melanosomes, more melanin and the dark melanin, fully dark melanin can be observed. Okay, so this is actually a maturation process of melanosomes. I will discuss about the tyrosinase function in my later slides. Maturing melanosomes. मेहताब साहब ये दरवाजा बंद करते हैं, दरवाजा बंद करते हैं. Melanosomes accumulate more pigments as they mature and move to the periphery of the melanocytes. Okay, so melanosomes accumulate more pigments as they mature and move to the periphery of the melanocytes. You have observed in this slide. In later stages like stage 3 and 4, they get more melanin accumulation. Okay, stage 2 is a young melanocyte and stage 4 is a mature melanocyte. Okay, the internal environment of the melanosome dictates how much pigment is made along the way. Okay, so definitely internal environment of the melanosome matters. So this is actually a melanin synthesis, which is important. This is actually a part of your physiology, how melanin produced. So first of all, you can observe a diagram here at right corner of the slide, in which you can observe the MSH word, alpha MSH. What is MSH? MSH actually a melanin stimulating hormone. Okay, so MSH, which is melanin stimulating hormone, binds with the receptor, which is MC1R. Okay, so melanin receptors which are present on the melanocyte surface. So keep in mind, MC1 receptors are present on the melanocyte surface. So MSH bind with the MC1R receptor. And then, you know, after binding with the receptors, whole cascade, whole pathway gets activated. Then initiation of synthesis occur in the melanocyte cell membrane. Synthesis in melanosome, the genes that are expressed protein. Okay, so the proteins and the genes which are usually responsible for the melanin biosynthesis are MC1 R. So MC1 are actually receptor protein which are present on the melanocyte surface. You can observe here MC1R protein, ASIP protein. I will tell you the function of ASIP protein later in my slides. Then TYR actually another protein which usually perform function in the melanosome, inside the melanosome. Then TRP1 and TRP2, both are proteins from tyrosinase. Let's discuss a few things about this. The hormone alpha melanocyte stimulating hormone, okay, interacts with the melanocortin 1 receptor, MCR1 receptor. I just told you, on the outside membrane of the melanocyte. As a result, cyclic adenosine monophosphate level increases. Okay, so in simple wording, when MSH melanin stimulating hormone binds with the receptor, then cyclic AMP inside the cell increases. So when cyclic AMP increase, then a whole cascade gets activated. Usually a cascade of reaction convert the amino acid tyrosine to the pigment eumelanin. So I will show you other diagram in my next slides where you can understand easily. So now just keep in mind cascade gets activated and tyrosine is involved in the pigment red and black pigment accumulation or formation. People with the dark skin have lots of eumelanin. Okay, people with dark skin, but people with light skin have pheomelanin. Okay, So more pheomelanin defines light color skin, and more eumelanin defines dark color skin, dark brown color skin. Okay, so few more things about the melanin biosynthesis. Tyrosinase is an enzyme, TYR, which is mentioned in this slide. Tyrosinase coded by the TYR gene. First of all, gene, you know, all proteins are as a result of expression of respective gene. So TYR gene is responsible for the production of tyrosinase enzyme. Okay. So this enzyme is involved in the conversion of thyrosin to melanin. Okay, so tyrosinase enzymes convert the thyrosin to melanin, and a few other proteins are also involved. Okay, TYR is a copper containing oxygenase, and leave this slide now. A TYR actually are of two types. Okay, so for example, nucleotide difference in the TRY gene correlates with the skin pigmentation. I have just told you, if TRY variants you can observe in European peoples, TYR is actually which type is TYR existing in the European people, so wo hai RS2733832 is associated with lighter skin pigmentation. Okay, so mean to say ke TYR also defines skin color. So variation exists in the TYR. So we say this is TYR polymorphism. Okay. Then there are two thyrosin related proteins, TRP1 and TRP2. TRP1 is the one is TRP, the other is dopachrome. Sometimes you will observe dopachrome word, dopachrome tetomerase. Okay, DCT, we can say DCT as well. And in other words, you can say TRP2. So there are two thyrosin related proteins. Okay, thyrosin related protein. Okay, tyrosinase. Now, what is the difference? Tyrosinase is an enzyme. Tyrosinase is an enzyme which convert thyrosin protein into melanin. And there are two types of thyrosin protein, TRP1 and TRP2. TRP2 usually known as dopachrome tetomerase. Now, this is actually a very important and easy diagram just to make sure that slides should be easy for you. Tyrosine hydroxylase enzyme converts the uh, into dopa and the dopacunine, and after few steps like leukodopachrome 5,6-dihydroxyindole, indole 5,6-quinone, then melanochrome forms, and melanochrome convert into melanin black polymerase. Here you can observe a tyrosinase function. Okay, so TRP1 and TRP2, TRP1 tyrosinase can convert melanin black color polymerase, and melanin black color polymers means melanin production. Okay, so if we say melanin black color polymers forms after a cycle, then we say melanin forms. And on the other hand, dopachrome converts into cysteine, and then melanin red polymers mean to say pheomelanin. Okay, here you can observe, I have just told you, pheomelanin are produce red color, and melanin produces black dark color. Okay, so here you can observe, if red polymers are formed, it means pheomelanin forms, and then in case of melanin black polymers, then you can say melanin forms. So both pigments decide skin color. Okay, in simple wording, in very simple wording, both pigments decides skin color. Okay, so if red polymers are high in number than melanin, then skin will appear light in color. If dark pigment melanin is more in skin, then skin will appear dark in color. Now, melanin deposition. Melanin deposit normally as well as abnormally or pathologically in our body. Okay, so in normal circumstances, where melanin deposition occurs, so you can say skin, brain, eyes, and abnormally, usually in common abnormal deposition of melanin is common in many neoplastic growth or tumors. Okay, so first of all, if we talk about the deposition of melanin skin, then we say ke, then we can say ke, as you have just studied ke, melanocytes scattered melanocytes are many numbers in epidermis. They produce dark pigment in certain acute inflammatory diseases. In certain acute inflammatory diseases, in certain acute inflammatory, it can be found in phagocytic cells. Okay, because phagocytes are in because macrophages or phagocytic cells are involved in the engulfment of excessive melanin. Then due to when phagocytic cells even take melanin or engulf or phagocytosed melanin, then melanin gets deposited into phagocytes as well. Then we can say it's a melanophages. Okay, so we can modify this word melanophages from macrophages, because macrophages are phagocytic cells. So when macrophages take melanin, then they become melanophages. Melanophages can also engulf other pigments and show unexpected staining result, because the melanophages, if other pigments are also there, like lipofuse skin, okay, so melanophages can also engulf lipofuse skin. And after staining reaction, uh, due to presence of mixture of pigments like lipofuse skin, melanin, staining, surprise staining reactions can be observed. Now this was the normal accumulation of melanin, but melanin accumulation can observe pathologically as well in our body, but there are diseases. When we say pathological accumulation, then you should aware that this is actually disease related accumulation or any abnormal accumulation. So in case of pathological, benign lesions, nevus or mole formation, this is actually a benign lesion in which melanin accumulation can be observed. On the other hand, malignant nevus are known as melanomas, which show the accumulated melanin. Okay, so there are two, benign lesion and malignant lesion. Melanin also found in hair follicles of dark-haired people. Okay, so when we talk about the melanin deposition in the skin, you should know that there are benign and malignant lesions as well. Okay, so in benign lesion, nevus, and in malignant lesion, melanomas are usually common, in which we can observe melanin deposition. If we talk about the deposition in eye, then normally in eye, melanin is present, where choroid, ciliary bodies, and iris. If you don't know about the anatomy of eye, then go first and study and revise the anatomy of eye, because a choroid, ciliary body, and iris are the part of eye. Okay, so normally melanin is present in the choroid, ciliary body, and iris of the eye. In retinal epithelium, there is any other unknown pigment is also present, which unknown pigment is slightly different from melanin, but we cannot say melanin is actually present in the retinal epithelium. Okay, so melanomas can occur in the eye, but rare. Means to say that pathological melanin deposition can also occur in eye, but it is rare. Then if we talk about the melanin deposition in brain, then first of all, where melanin is present, so substantia nigra is a site where normally melanin is present, and melanin is present in very large amount, so that mesencephalon appear dark from both sides. Okay, so these are actually anatomical parts of brain. So if you don't know this now the substantia nigra, mesencephalon, then go and study atlas of anatomy first. Okay. Okay, substantia nigra and the mesocephalon are two sites where normally melanin is present, and melanin is present in normally in very high amount. Okay, dark area in arachnoid covering of human brain. Dark area arachnoid covering of human brain is also arachnoids. What is arachnoid covering? You should know about the anatomy of brain. Okay, then they appear sooty appearance. Okay, sooty specific appearance you can observe easily. Then identification of melanin. This is important apart from lab point of view. If you are sitting in a histopathology lab and skin sample just received, and you just uh, uh, pathologist demands from you which stain would you like to do for melanin identification and to check the accumulation of melanin. So there are two stains: Mason Fontana's method of melanin and the Shimuras reaction. Okay, there are two common stains. Uh, Mason Fontana actually a method first described and then modified by the Fontana and Mason respectively. Fontana in 1912 first discovered this method, and Mason in 1914 just modified this. Shimuras reaction is created by Lilly's is actually discovered by the Lilly in 1954. Okay, so just due to the just due to the availability, on the basis of availability or price, we decide which reaction, which method we can apply. Both can show best results. Okay, so if we talk about the Mason Fontana method, another important thing in UHS, in MCQs, first of all, in MCQs, examiner ask about the results of staining methods. Okay, interpretation of staining methods means to say if you did Mason Fontana method, to what was the result? Okay, and in SCQs, method, principle, or these things are usually asked by the examiner. So in first of all, fixation tissue, in which fixative tissue should be fixed. So formalin is the best fixative. Okay, formalin is the best fixative. Chromate and mercury chloride should be avoided because these chromate and mercury chloride can react with the Mason Fontana uh, reagents, and it will not give proper results. First method is take test tube and control section of distilled water. Means to say, first of all, take section, test and control section of distilled water, treat with the ammonical silver solution. Means to say, you have a section on the glass slide. First of all, take a glass slide on tissue is present to the distilled water, then after after distilled water, uh, treat slide or section with the ammonical silver solution in the copelin jar. Okay, uh, 30 to 40 minutes if temperature is 56 degrees centigrade of ammonical silver solution, or if solution is at room temperature, then for overnight. Means to say, treat its section with the ammonical silver, and other conditions are also mentioned. Wash after treatment with the ammonical silver solution, wash well in the several changes of distilled water. Mean to say, wash well. You have to remove completely as much as you can through by the distilled water. Treat section with 5% aqueous sodium thiosulfate for 1 minute. Okay, so after removal of excessive ammoniacal silver solution with the distilled water, you have to treat the section with 5% aqueous sodium thiosulfate. Then wash the section with tap water for 2 to 3 minutes just to remove excessive stain. Okay, then lightly counter stain with 0.5% aqueous neutral red. So now counter stain, you will apply counter stain, which is a commonly for 0.5% aqueous neutral or 0.1% aqueous nuclear fast red. Okay, for 5 minutes. Okay, so apply counter stain for 5 minutes and it will be 0.5% aqueous neutral red, commonly available. Rinse in distilled water. So after counter stain, wash the slide or section with distilled water and let the slide dry and clear. And after the after dry, you can mount the slide. And after mounting, you will observe the slide under microscope. So what will you observe in the microscope? So here are results of Mason-Fontana method. Melanin accumulation will appear dark and brown in color. Dark brown in color. Okay, so melanin, argentafin, and chromafin. These pigments usually black appear black in color. 5 से 6 मिनट में black in color. Okay, so in case of but in red, baki nuclei of cell appear usually red due to counter stain. Okay, so you can observe in diagram a dark color shows the presence of melanin, and the pink color or red color is showing the nuclei of the cell. Then another technique is Shimuras reaction. In Shimuras reaction, 10% buffered neutral formalin is used as a fixative, means to say there is no special fixative requirement. If a tissue is fixed in the formalin, normally as we do in our routine fixation or in our routine histopathological exam, first of all, we will fix the tissue in 10% buffered neutral formalin. Then we will take this section from dead tissue. First step is take test tube and control section to distilled water. Okay, so first of all, we will apply control and test. Second, treat section with the ferricyanide solution. Now you can observe there is a difference. There is a slightly expensive thing. Ferricyanide, ferricyanide or ferricyanide is actually not a very light solution. That it can be dangerous. So that's why we usually we perform according to the condition. We choose reaction or staining method according to safety, according to price, according to easily availability, or etc. Okay, so second step is treat section with the ferricyanide solution for 5 to 10 minutes. Okay, so wash well in running tap water for several minutes to ensure that all the residual ferricyanide is completely removed from this section. Okay, now after washing, lightly counter stain 0.5% with 0.5% neutral red. Okay, for 5 minutes. Then after dehydration, you will mount this light and observe under microscope, and you will get these results. In Mason-Fontana interpretation, you can observe dark staining, but in Shimuras reaction, you will observe melanin with blue color. Melanin with blue color. Okay, so here you can observe blue color accumulation in the tissue. Okay, central slide is actually a slide from muscle where you can observe different melanin deposition. Okay, so this is actually a interpretation of reaction, and most